Hello viewers, today we will be studying mannose binding lectin pathway. Now what is this mannose binding lectin pathway? This is one type of a complement activation system. This is how complement system gets activated. I have in my previous class told about classical way of activation of pathway. So that was called classical pathway of complement activation. This time we are reading about mannose binding lectin pathway, one of the other way by which complement can get activated. So what is this? This mannose binding lectin is a family of colectins, colectin 11 and even serum phycolins. This is present in serum. All these are synthesized in liver, present in serum, and they have collagen and carbohydrate recognition domains. That means these mannose binding lectin, that means sugar and protein, have the ability to recognize the pathogens sugar and N-acetyl groups which is present on pathogen. So they have that pattern recognition receptors, or you can say carbohydrate recognition domains. So they can recognize the carbohydrate pattern on pathogen and even the sugar pattern on pathogens. So they binds to sugars on or N-acetyl groups on the pathogenic organisms. They can even recognize abnormal, that is aberrant glycocalyx pattern on apoptotic cells. Cells, they have a different kind of cytoskeleton in the system, in the system, present in the system. They can, these mannose binding lectin molecules can determine those cells also, and they can determine the cells which have some not normal composition, that is they have become tumoral cells. So this mannose binding lectin molecule colectins, they are the group of colectins which are synthesized in liver and present in serum. They have the ability to recognize all those pathogenic and apoptotic cells as well as tumor cells. And when they recognize those cells, because they have pattern recognition receptors on them, and when they recognize such type of cells, they go and bind to those cells directly. Once they go and bind to that cell directly, they activates MBL associated serine proteases, which are MASP1 and MASP2. These two types of MBL associated serine proteases which are present, and when they get activated, they cleave C4 to produce C4A and C4B, as we have seen in classical pathway. In classical pathway, complement system 1 was activated by serine proteases. In this mannose binding pathway, what happens? It directly goes and binds to this group of colectins, go and bind to pathogen or apoptotic cells or tumoral cells. They directly bind to pathogen, and from there they activate MABL associated serine proteases, certain serine proteases which gets activated once they bind to pathogen. And when these gets activated, this MBL associated serine proteases 1 activates serine proteases 2, and that cleaves C4 to produce C4A and C4B fragments. C4B again goes and directly attaches to pathogen. So what all are attached to pathogen? First, it was mannose binding lectin protein which was attached to pathogen. Second, because they had the domain to know that this is the pathogen, they have the recognition factor, so they directly recognize and associate, and they bind. Once they bind, the MABL associated serine proteases get activated, and that pulls the complement C4 and breaks up, cleaves it into C4A and C4B, while C4B directly goes and attaches to pathogen itself. And then this C4B, when it's bind to the pathogen, MABL associated serine proteases cleave C2 also, and it cleaves C2 into again two fragments, C2A and C2B, as we have seen in classical pathway also. And when C2B also goes and attaches to the immobilized pathogen where C4 is also immobilized, it goes and attached to pathogen where C4B is also immobilized and attached to the pathogen, binds to the pathogen. So now what complements have gone and attached to the pathogen? It is C4B and C2B. So these two gets associated and binds, and then these C4B and C2B. Just remember, C4B and C2B together activates C3 convertase. C4B and C2B. This is very important to remember which complements activate C3 convertase. Once C3 convertase is activated, what will happen? C3 convertase, as the name signifies, this C3 convertase will act on C3, and then it will cleave C3 into C3A and C3B. Again, C3A and C3B was the complement which was the turning point, or which called others. This was the first enzyme C3 convertase in the classical pathway also. Once C3 convertase gets activated, mannose binding lectin pathway also follows the same pathway that was followed by classical one. Only thing was, classical was activated while antigen antibody complexes were formed. In this case, this mannose binding lectin, this is the group of colectins which directly go and attach to pathogen, and then they activates complements. So in this case, there is no role of C1. That is the difference between classical pathway and mannose binding pathway. There is no role of C1 in mannose binding pathway, since mannose binding lectin pathway is a central molecule in this, because it is having the pattern recognition receptors. It recognizes the pathogens, sugar or N-acetyl group. It even recognizes the apoptic cells. Why it's able to recognize apoptic cells? Because of the aberrant, that is the change structure they are having. This mannose binding lectin can even recognize the tumoral cells because they are not having the normal structure. So this mannose binding lectin pathway is in this way different from classical pathway. But once C3 convertase get activated, this mannose binding pathway also follows the same path as it is followed by classical pathway. A classical pathway I have taken in the previous lectures, and I will be putting the link in the description box. So who all are interested can go through classical pathway also. This complement pathway is complement system is a part of immune system where innate immunity and adaptive immunity can talk to each other through this complement pathway. So this complement pathway, once C3 convertase is formed, this C3 convertase will cleave C3 into C3A and C3B. Again, the C3B will attach to C4B and C2B, which were before present on the pathogen, which were bound to pathogen. And this, when C3B also goes and bound to C4B and C2B, this will activate C5. C5 convertase, another enzyme, C5 convertase. And as it is called C5 convertase, it will act on C5 and break C5 or cleave C5 into C5A and C5B. This C thing may make you confused, but remember this C is the complement protein which were discovered, and according to their discovery, they were named 1, 2, 3, 4, and 5 whenever they were discovered. Later on, it was come to know that C4 and C2 has a role together, and then they C4 and C2, that is also B fragments. These fragments are the larger fragments. The smaller fragments too have a role, that is C4A, C2A, even C3A and 5A, all they have a role. And they are the basically 3A and 5A are the anaphylotoxins. That in what is the role of anaphylotoxins? Anaphylotoxins increase the inflammatory response, and they can directly activate basophils and mast cells to produce inflammatory response. They can directly go and influence. So C3A and C5A, they diffuses and they influence mast cells and basophils to cell mediators. So they also have a role in controlling the infection. While C5 convertase is formed, it is a protein complex consisting of C4b, C2A. C2A was formerly, but now it's like C2B. So it's C4B, C2B. Please correct it. It is C2B and C3B. So this C5 convertase, when cleaves C5, it initiates activation of, as we have seen it in case of classical pathway, that C6, 7, 8, 9 are activated, and C5B to C9 together it goes and attaches to the, as you have when I started mannose binding pathway, I started that mannose binding lectin goes and binds to pathogen directly. When it is bind to pathogen, at that time mannose binding MBL serine proteases gets activated, and one gets like serine proteases one gets activated, and then serine proteases one makes two MABL2 activated, MABL serine protease 2 activated, and this 2 1 goes and activates C4 and C2, and C4 and C2 together, be part of these together form C3. And once C3 is formed, it's like a classical pathway itself, and then this all is bind to the pathogen, and then this pathogen cell wall is again bind, and this pathogen is killed by membrane attack complex or terminal complement complex is formed. And this way, these mannose binding pathway or classical pathway makes the same, they kill the pathogen, and but the paths are different because they are following different routes and gets activated through different processes. So those who want to know how classical pathway gets activated, I'm putting a link, sending a link, and you can see that video too. Now, the biological effects of MBL pathway, just summarize, here I am summarizing the biological effects of MBL pathway. That is, one, I have already told you that complement activation takes place. Another thing is opsonophagocytosis. Now, opsonization is just immobilizing immobilizing and facilitating the clearance of infective organism, while sometimes they directly hand over all this that they have attacked to macrophages and dendritic cells also to initiate phagocytosis. So MBL pathway can hand over the infective organisms, pathogens to macrophages and dendritic cells, and thus initiate phagocytosis. Then modulation of inflammation, that means the inflammatory molecules, anaphylotoxins are secreted in this case, and these modulate the inflammation. That means inflammatory responses are also modulated by this MBL pathway. While recognition of altered cell structures also take place because they have recognition domains, they have recognition receptors. So if there is some changes in the cell itself, our own cell, that is the body cells, immune system is there to protect us. So if our own host cell has got modulated and have become their cell wall have changed, and their cell membrane, not cell wall, basically it's in the case of our host cell, human host cell, cytoskeleton of the cell or cell membrane have somewhat altered. So the role of MBL pathway is to clear them also. So this was all in today's lecture. Thank you very much.